Hello everyone. Today I'm going to be talking about fertilization and the early development of the human embryo. Okay, give me a second. Let me just share my screen. And my screen should be visible to you as about now. Okay, so what is fertilization? As you know, fertilization is when the male nucleus combines with the female nucleus. Essentially, the gametes between the sperm and the oocyte collide and form a union, specifically in the fallopian tube. As you know, the entire backstory of this process, it's when sperm gets developed and deposited near the crevices of the vagina. And from there, it makes its way up to the fallopian tubes where it combines with the human egg. So I like to divide fertilization into six main stages. The first stage is the penetration of the corona radiata. It's essentially where the head of the sperm, filled with a bunch of enzymes known as acrosome, um, loosens up the structural integrity of the corona radiata, which is the outer layering of the egg cell, right? And this essentially provides a better pathway or better access for the sperm to reach the female pronucleus, right? The next stage is where after the corona radiata gets penetrated, the zona pellucida, which is another inner layering right underneath the corona radiata, it too will get digested. So the acrosomal enzymes again continue to get released and digest its way through the zona pellucida. Okay. Stage three is when the cell membranes begin to of the sperm and the oocyte begin to fuse. So as these two cell membranes, as they're about to meet, the cell membranes begin to fuse, and when they do so, it causes the cell membrane and something called a perforated mitochondrial sheath to be left behind, right? So the mitochondrial sheath, which provides the double membrane outer layer covering for mitochondria present in the sperm, will be removed and left behind and not involved in the fusion process between the oocyte and the sperm. While this stage is happening, the essentially cortical granules from the oocyte get released, and these cortical granules hold a very high importance because it causes the corona radiata to shift chemically, which causes no other sperm to penetrate through the corona radiata. So this prevents multiple sperm fertilizing one egg. Right before the nucleus of the sperm meets the pronucleus of the egg, the oocyte actually just finishes or just completes its second meiotic division. So now it's officially mature enough for fertilization. And during fertilization, what essentially happens is the nucleus of the sperm enlarges, which forms the male pronucleus. The nucleus of the egg enlarges, which is present already and is mature now, and they both combine. So when these both combine, the tail actually of the sperm, which has been a great help so far because it's been involved in the propelling of the sperm through the corona radiata and the pellucida into the region where the pronucleus is held. So that tail will now eventually degenerate. Right? So when it degenerates, the both the nucleuses fuse together and form a chromosomal arrangement where you have 46 chromosomes present inside something called a zygote. Okay, something called a zygote. So once the zygote gets formed, fertilization is officially over, and early development of the infant begins. Okay, so early development is actually split into a couple of days, which are very, very important for you to remember. The first one is something called cleavage of zygotes. So the first days, which is somewhere around 24 hours to 30 hours, is essentially the stage when the zygote continues to rapidly increase in mass. Right? So when I say increase in mass, I'm basically referring to mitotic cell division. So the zygote eventually changes to something called blastomeres. So they multiply in number of cells, and this increases the mass. So once it reaches a stage where there are at least 16 to 32 cells present inside the blastomere, we then no longer call it a blastomere, but we refer to it as a morula. Right? So morulas basically have 16 to 32 stages of cell mass present inside of it, and the outermost layer, if you can see over here, is lined by a green a green representation, this outermost layer is then going to be called a trophoblast. Right? Trophoblasts are very important in terms of pregnancy, which we will deal with later. And when we talk about implantation, so not to worry about that. But essentially, after the morula continues to enter the uterine cavity, it then gets to be called something called a blastocyst. Right? So when it penetrates and it enters the uterine cavity, it becomes a blastocyst. Why? Because the the region where it's getting implanted is something called the blastocystic cavity. Okay, so from days 4 to 6, the blastocyst basically makes its way down to the uterus and gets itself ready for implantation. And in this time, it continuously rapidly multiplies itself by 2 in order to increase itself in size. Okay, so at the end, on day 6 or 7, when the blastocyst eventually attaches itself to the endometrial lining of the uterus, it then allows its trophoblast, which is the outermost layer, to penetrate inwards towards the uterus wall. And when it does so, it forms something called a cytotrophoblast and a syncytotrophoblast, two very important things when it comes to the maturation of the blastocyst. Okay, this is what it looks like to give you guys a better understanding. The morula occurs over here, and as it continues to divide, it forms a blastocyst. So the blastocyst is outlined by your cytotrophoblast and the syncytotrophoblast. And this is basically the case of what it looks like once implantation already occurs. Okay, that's what it looks like. And this is a better representation, including the uterus wall. Okay, so this is representation with the addition of the uterus wall, and it gives you a better understanding of how exactly the trophoblastic outer layer makes its way into the uterus.